To begin with, psychopathy is understood as a personality disorder that is comprised of deviant and defiant social behaviors. Psychopathy is marked by several traits. Some of these traits include lack of empathy, guilt, conscience, or remorse, shallow experience of feelings or emotions, and the irresponsibility of accepting one's actions. For centuries, on both international and national scales, psychopaths have terrorized and tortured populations. In both examination of world history, that is ancient and modern, the faces to these psychopathic crimes have been white. The aim of this study is to detect and explore the genetic influence and presence of psychopathic behaviors in white Americans. This study analyzes in depth the racial trend of psychopaths in American history to investigate, understand, and analyze the relationship of genetics to race and psychopathy. Research suggests that some traits of psychopathy are predicted by significantly genetic factors and can often appear before the age of 10. There are many different factors and influences that lead to psychopathy. Primary psychopathy is suggested to be caused by genetic influences, where secondary psychopathy could be a result of traumatic childhood exposure. This research models its thesis after the lynching of Mary Turner. The lynching of Mary Turner is a case in which a white mob terrorized the Black people of Brooks County, Georgia, in response to the murder of a plantation owner. This lynch mob went on a rampage that killed an estimated 13 people, and out of those 13, Mary Turner contested to her husband being one of the slain. Heartbroken and enraged, Turner threatened to have warrants out for the men who killed him. The Brooks County lynch mob response to Mary Turner's warrants were atrocious. Once the lynch mob found Turner, they hung her upside down from a tree by her ankles, poured gasoline on her body, and then burned off her clothes. One member of the mob then proceeded to cut open her stomach and watch her unborn child drop to the ground, where they reportedly stepped and crushed the baby's head 11 times with their heel. Her body was then riddled with gunfire from the mob, and later that night, her and her baby were buried 10 feet away from where they were murdered. The grave was marked by a whiskey bottle with a cigar stuffed in its neck. After learning about the lynching of Mary Turner and really conceptualizing and imagining the mob committing those crimes, led to the research question, are white Americans genetically predisposed to psychopathic behaviors? Which then would mean if psychopathy is a prominent gene in white Americans, then various genetic tests will prove so. For this study, we chose to use the casual comparative research design because we are aiming to identify the cause and effect relationship between psychopathic genetic composition and expression in white Americans specifically. We will use the retrospective casual comparative research design because the effects of white psychopathy in history has gone unquestioned for many years. The retrospective design model looks at the effects that have already occurred versus the cause and seeks to explain the relationship between cause and effect. Although the cause of psychopathy still remains very unclear to researchers, there's accumulating literature that indicates genetic influences promote the development of psychopathic traits. Although 20% of the prison population in North America are classified as psychopaths, the general adult population of psychopathy is only about 1%. Psychopathy is a developmental disorder marked by an increase in antisocial behavior and emotional deficits. Jeffrey Dahmer, Ed Gein, Ted Bundy, Gary Ridgway, Dennis Rader, John Wayne Gacy, and Ed Kemper are not only serial killers that are identified as psychopaths, but American white men. A great amount of research points to psychopathy being genetically determined more so than environmentally influenced. Although the topic of behavioral genetics remains a controversial topic, some psychopathic behavior may be grounded in genes. There's a case of the X-linked MAOA gene. Gene is associated with general antisocial tendencies. Psychopathy and race. Race has been used as a variable to group and compare results reflected in data collection and psychopathy research. Test results seen in the Minnesota Multiphasic Personality Inventory study concluded East Asians scored lowest, followed by whites, Hispanic, and then African Americans. Result samples from the PCLR test indicate that it was created to serve Caucasian offenders over other races. Samples that were found in Caucasian offenders were not located in non-Caucasian offenders. Ethnic differences in levels of psychopathy. Although there isn't a lot of research regarding the relationship between psychopathy and race, a dense amount of rhetoric points to psychopathy as a prominent disorder in African American communities. However, these sources do not consider the pre-existing disparities in institutions in Black communities, for example, poverty, that would point to the display of psychopathic tendencies. Meta-analytic evidence specifies that psychopathy is a weaker associate of violence recidivism among more ethnically diverse samples of juvenile offenders comparatively to white samples. There's a considerable amount of heterogeneity when it comes to the observation and report of psychopathic traits amongst Black and white groups. In our design and methodology, 2 groups were chosen. The first group identifies as white American and has a family history of psychopathy. This is our experimental group. Our second group is our control group. They also identify as white American, but they do not have a family history of psychopathy. Recruitment for our participants will be done through a flyer-like forum that will connect itself to websites of people with this type of demographic. 100 participants will be chosen per group from 5 different states. Within those 5 states, 20 individuals will be chosen. The 5 states were chosen due to the presenting data that these states have the highest frequency of psychopathy. Our independent variable for this experiment is the psychopathic genetic presence in white Americans. Our dependent variable is the psychotic behavioral expression. The measure that will be used for this experiment is the Hare Psychopathy Checklist Revised, aka the PCLR test. The PCLR test consists of a 20-item symptom rating scale that allows qualified examiners to compare a subject's degree of psychopathy with that of a prototypical psychopath. Since this is a Likert scale test, a total of 20 items are measured throughout this process, given scores 0, 1, or 2 based on how effective they apply to the participant being tested. The PCLR provides a total score that indicates how closely the test subject matches to the perfect score that a classical or prototypical psychopath would rate. A prototypical psychopath would receive a maximum score of 40, while someone with absolutely no psychopathic traits or tendencies will receive a score of 0. A score of 30 or above qualifies a person for a diagnosis of psychopathy. In a causal comparative design, the test is used to determine if there is a significant difference between the means of the 2 groups, because we assume that the dependent variable fits both groups automatically. The procedure for this study would be to inform the participants of the study being conducted upon arrival on the psychopathy gene in white Americans and asked to complete consent forms. A questionnaire will then be given to confirm if they are genetically predisposed to psychopathy and their demographic information, then will be separated into the 2 groups based upon these questionnaires. Participants will then be separately taken to receive an fMRI and will be tested for the monoamine oxidase A enzyme. The purpose of the fMRI is to scan for damages found in the amygdala and prefrontal cortex that could also be a cause for certain types of psychopathic behaviors. After the testing is completed, participants will be compensated with a $20 gift card and given further information regarding the rest of the study. In this causal comparative design, the independent variables' effect on the outcomes will be seen by the similarities and differences measured in the 2 groups, and these results will be compared to find the final results. Data analysis includes the data will be analyzed using descriptive and inferential statistics. T-tests will be used as measures of inferential statistics that determine whether or not the statistical difference between the 2 means are significant.
The standard deviation will be used as a measure of descriptive statistics that look to measure the dispersion of the data set relative to the average.